Students, continuing our class with the kinetics of product formation. As I said earlier, the kinetic model suggested by Lutking and Pirate was originally designed for gluconic acid production using the bacterial cells, and it asserts that the product formation depends on the microbial growth during the fermentation process in a linear fashion. So, what I told in my previous class, I told you that when we are speaking about the kinetics of product formation, we use this LP model, and this model was designed based on the gluconic acid production by a particular bacteria, and it showed a linear fashion of curve during the fermentation process. And this equation has been applied to characterize the product kinetics. Maybe it can be your monoculture cultivation of filamentous fungi when you're using submerged fermentation or solid state fermentation. You can use it even to identify, okay, the model was suitable to identify the kinetic constant of mixed fungi during your submerged fermentation. So both, whether it is a monoculture fungi or mixed culture fungi during your fermentation, whether it is a submerged fermentation or solid state fermentation, they were able to use the model to predict the kinetics of the product formation. So the model what you're seeing here, okay, in this picture, ds by dt, okay, is equal to minus gamma dx upon dt minus lambda x. So this is also the using the same LP model. This is also being predicted here. What is the differences? See here, in previous class what we studied, we told this is the LP model, okay, for the product formation. This one. So now the substrate consumption, on the other hand, can be depicted with the same model, and it is given by this. So what do we understand by each of this terminology? So gamma means milligram per gram, and lambda is milligram per gram, are the Lutking Pirate equation parameter for growth and non-growth associated constants for substrate consumption. So we told that the LP model is used for the mixed growth products formation. Okay. And then there we told it is alpha and beta. Okay, if you remember that. So growth associated and non-growth associated. Here we use another terminology, gamma and lambda, which is growth associated and non-growth associated. The mathematical equation described by this LP that involves a plot of specific growth rate mu versus product formation produced a straight line whose slope is growth associated coefficient alpha, and the intercept is non-growth associated parameter beta, was used to observe the protein synthesis by the fungal mixed culture. So what this entire sentence means to tell is, by using this equation, the investigators, what they did was they tried to plot a graph, okay, which involves the specific growth rate, that is the mu, okay, versus the product formation produced a straight line. I'll show you here in the next slide, then you can understand it. But meantime, you try to grasp this what I'm telling. Whose slope is growth associated coefficient alpha, and the intercept means the where it meets the zero, okay, is non growth associated parameter beta was used to observe the protein synthesis, how protein synthesis occurs, okay, when a mixed culture of fungal population was involved. So when they used this equation and when they conducted the experiment, what was the substrate they used? They used a mixture of the fungus. And what did they want to observe? How the protein synthesis has been formed. And they plotted a graph. When they plotted the graph, they got the relative coefficient value of 0.988. Look at this. So the specific growth rate per day, okay, the specific product formation using that equation when they conducted the experiment and when they plotted the graph, okay, they got a relative coefficient of 0.988 and it was a straight line graph. And here also you can see specific growth rate per day and specific substrate consumption rate. Okay, it also gave a straight line graph with an r squared value of 0.9384. This clearly shows that how well the graph can fit. I mean, the model can fit for this particular experiment. You can see this whenever the r squared value is zero point 0.9 and even for some extent zero point 0.8 also is accepted, but zero point 0.9 and above, whenever you get a r squared, you can say that there is a perfect fit of your model or your the graph is perfectly fitted. That is, I'm considering the specific growth rate of my microorganism and how the substrate is being consumed. Okay, here what I'm considering the specific growth rate of the organism and how the product is being formed. Both I'm considering the growth rate of my organism, how the substrate and the product are being, the substrate is being consumed and product is being formed. You can see that when we plot a graph, we got an what relative coefficient which was very good. Here you can see it is really a very good value we have got 0.988. Almost when it comes to 1, it is a perfect fit. Okay, but rarely we get a perfect fit. What I mean to tell is when we are doing experimentally, okay, so that is the entire meaning of this sentence. So the rate of formation of product directly relates to the rate of growth. This is what we understand by the model. Okay, the rate of formation of the product is directly related to the rate of the growth of the microorganism, and we saw that with the help of this graph. An important characteristics of modeling is assumption of homogeneous or heterogeneous condition. So whenever you are considering your modeling, you have to consider even how what is the condition of the environment, whether it is homogeneous or it is a heterogeneous. So homogeneous system is related to a single continuous phase. What we mean to tell is when you're running your bioreactor, okay, in most cases, bioreactors are described as a single liquid phase because liquid is what there we are adding a nutrient media there. Okay, but we have also spoken about in our previous classes known as if there is a biofilm is included in the study, then you cannot say it is a clear liquid phase. It is a solid or a semi-solid phase. Okay, on the other hand, when we are saying heterogeneous system, if your bioreactor is in a heterogeneous system, what does it mean? That is, it is involving two or more continuous phases, and there is an interaction between these two or more continuous phases. These continuous phases, okay, it can be your liquid or solid, or it can be your semi-solid, and it can also involve your gaseous phases. Okay, so for example, solid state fermentation, wherein we spoke about what we use the substrate. Okay, as such we used it. That is what we spoke about the solid state fermentation. That is, it is an heterogeneous. There is no one phase there. Hence, it is a heterogeneous phase. Whereas if it contains only one phase, it is called as a homogeneous. Okay. Suppose say there is this liquid, solid or semi-solid and gaseous phase all present, then it is heterogeneous, but it is complex heterogeneous.